Hello, welcome to the In Vivo Imaging Knowledge Sharing Video Forum. I'm Dr. Alexandra Delille and Director of Technical Applications at PerkinElmer for In Vivo Imaging. If you have questions and want to explore further, feel free to email me at alexandra.delille@perkinelmer.com. Today we will discuss the advantages and methods of in vivo fluorescence and Cerenkov imaging of antibodies, proteins, and peptides. Optical imaging is a valuable methodology for preclinical pharmaceutical development. It is low cost, high throughput, with high sensitivity, and translational to human medicine. Let's start with some examples of peptide, protein, and antibody imaging. Here you can see an example of antibody tumor targeting. A TD tomato expressing PC3M tumor, represented in orange, is targeted with a fluorescently labeled therapeutic antibody, represented in blue. HER2-Sense is a fluorescently labeled humanized antibody based on the commercial therapeutic antibody trastuzumab that specifically targets and binds to HER2-positive tumors such as the SCOF3 ovarian cancer cell line. MDAMB231 cells were implanted by intracardiac injection in nude mice. Multiple metastases, including bone mets, were formed few weeks post-implantation of cells. The image on the left shows the bioluminescent signal. Herceptin conjugated to xenolide 750 was injected intravenously, and the fluorescence image shown on the right was taken 24 hours post-injection. The antibody conjugated to the dye shows great targeting to the metastatic sites. The Rediject integrin 750 probe is a fluorescently labeled cyclic RGD peptide that binds alpha-V beta-3 integrin, enabling non-invasive fluorescence imaging of, for example, U87 Mg luciferase tumors in nude mice. Here is an example of a pharmacokinetic cross-validation of a dual-labeled Cy55 and copper-64 Naughton peptide probe in a murine human tumor xenograft model. Plots of tissue ratios correlated well over time. This data is published by Kimura et al. In Bioconjugate Chemistry. In vivo optical imaging methodology, what does it take? Labeling of peptides, proteins, and antibodies with a fluorescent dye is easy. You can label any peptide, protein, or antibody with an easy-to-use kit. Near-infrared fluorochromes specifically optimized and validated for in vivo imaging are available. You can choose from amine-reactive near-infrared fluorochromes for labeling via an NHS ester linkage, thiol-reactive near-infrared fluorochromes for coupling via malamide chemistry to label free cysteines or thiol groups. Non-reactive control dyes of the same wavelength are also available. In order to determine biodistribution, it is important to design a study with the correct treatment and control groups. Traditionally, we have a blank control group, a non-reactive dye-only group, a block target group, the fluorescently labeled antibody of interest group, and a non-specific IgG labeled antibody. In 2D settings, both dorsal and ventral images are taken. This data is from the group of Dr. Zhu et al. Published in Molecular Pharmaceutics. The same mice are imaged longitudinally at various time points up to 96 hours. Regions of interest in the tumor and liver or kidneys are quantified and data is plotted over time. In vivo methodologies still need ex vivo validation. Tissue fluorescence can rapidly be quantified and validated in ex vivo tissues. Simply take your last in vivo image, necropsy the tissues, and image individual organs ex vivo with the in vivo optical imaging system. A challenge faced in monoclonal antibody preclinical development is whether the antibody is specifically binding to the relevant target in the tumor tissue. Although we can observe accumulation of the fluorescently labeled antibody in the tumor with in vivo imaging, we still need ex vivo validation. With the Nuance spectral and mixing microscope camera, we can not only see where the fluorophore is inside the tumor in ex vivo tissues, we can also co-localize the antibody binding to the antigen with up to 5 molecular markers simultaneously within a single tissue section. Serum stability is an important assessment in the therapeutic antibody development decision-making process. The lab chip GX2 offers a high-throughput analytical technique with very good precision and sensitivity. Fluorescently labeled antibodies can readily be analyzed from blood. There is no need for gels, and electropherograms give a reading in just seconds from a microfluidic chip. Sensitivity of fluorescence imaging. Fluorescence imaging sensitivity in deep tissues is greatly enhanced by methods of transillumination and offers picomole sensitivity. One can detect 0.4 picomoles of 800 nanometer quantum dots deep in the lungs of a mouse. Translational imaging. Optical imaging of radioactive probes by Cerenkov light. Prior to clinical trials, an antibody can be tagged with a clinical radionuclide. For translational purposes, the distribution of this radionuclide can be screened in mice using an IVIS in vivo optical imaging system which has the sensitivity to detect the Cerenkov light emitted by both beta plus or beta minus radio tracers. Cerenkov light imaging data has been found to correlate well with immunoPET data and offers the potential to conduct rapid, low-cost, and high-throughput screening of novel radio tracers in vivo. Cerenkov light is visible light produced by charged particles. Both beta plus and beta minus particles generate light, and therefore various PET and SPECT radionuclides can be detected by optical imaging. In a study conducted by Drs. Ruggiero et al., medical isotopes were imaged by PET and Cerenkov, and both the qualitative and quantitative data were found to strongly correlate. Biodistribution of fluorescent antibodies, proteins, and peptides can be visualized in 3D tomographic reconstruction. By means of transillumination imaging, 3D ROIs, and tomographic reconstruction algorithms, we can quantitatively assess whole body biodistribution of fluorophores in 3D. The quantitative aspect is confirmed by strong correlation between in vivo and ex vivo tissue fluorescence quantitative data. Here is another example of a 3D reconstructive representation of Sci-Fi-5 biodistribution to deliver in co-registration with micro-CT. As a final note, I would like to quote supportive references from the literature. Kimura et al. Published, dual modality near-infrared PET imaging agents are promising for clinical applications in both deep tissue imaging and for image-guided surgical resection. Further stating that copper-64 and Sci-Fi-5 uptake plots correlate well, demonstrating pharmacokinetic cross-validation. Fukukawa et al. Found that results of in vivo biodistribution of a monoclonal antibody assessed by both radionuclide and fluorescence imaging modalities were very concordant. Dr. Sun et al. Simultaneously evaluated two antibodies, each labeled with a fluorophore of a different wavelength, in one and the same tumor mouse, allowing to assess potential interactions that can affect pharmacokinetics and potential side effects. For Drs. Dean et al., the high throughput, low cost, short anesthesia, and lack of ionizing radiation is a great advantage. When looking at the advantages of Cerenkov light imaging, Dr. Robertson from Millennium found the statistically significant correlation between PET and Cerenkov and the tenfold increase in throughput capability of Cerenkov light imaging versus PET imaging to be a significant plus. Drs. Ruggiero et al. Also stressed the advantage of the rapid, low-cost, high-throughput screening methodology of Cerenkov light imaging. Thanks for viewing this knowledge sharing presentation. Stay tuned for more videos. If you have questions, feel free to contact me by email at alexandra.delille@berkenelmer.com.